Thank you to everyone for being here today, and to Joan Silk and the UCLA Center for Behavior, Evolution, and Culture for inviting me here to share some highlights from my research on non-human primate behavioral ecology and endocrinology. My research interests lie in the intersections between animal behavior, ecology, and physiology, and much of my research uses multidisciplinary approaches that combine the observational, non-experimental approaches of field-based behavioral ecology and endocrinology to better understand the sources and consequences of variation in animal social behavior among individuals and groups in natural populations. During the past 2 decades, advances in non-invasive techniques to measure excreted hormones in animal waste, like in feces and urine, have enabled researchers like myself interested in hormone behavior relationships in free-ranging animals to study these topics without disruptions to the animal's daily routines. In my research, I use non-invasive hormonal sampling techniques to both assess potential determinants of animal social behavior and to evaluate potential physiological consequences of behavior. And this field-based approach allows me to study hormone behavior interactions within the selective environments under which these processes likely evolved. However, they are not without their drawbacks, as observational non-experimental studies of hormones and behavior are of necessity correlational in nature. Studies like mine cannot tell us about cause and effect relationships between hormones and behavior, but what they will do is they can provide insights into hormone behavior relationships. They can be conducted on a greater variety of species than is possible in the laboratory, and then finally they can help to confirm or in some cases refute or deny hormone behavior relationships that were previously established in captive animals. Much of my past and present research has focused on wild populations of primates in Kenya, Vietnam, and Rwanda, and in Ethiopia today. I earned my PhD in ecology and evolutionary biology in 2006 from Princeton University, where Joan and I recently, and Nguyen, I believe you were there as well, recently returned to celebrate recently returned to celebrate my PhD advisor, Jean Altman's retirement from service and teaching, but not from research. To celebrate her impending retirement. Jean Altman's now 40-year-long study of the yellow baboon population has been ongoing since 1971, and I was really fortunate to be able to work at Amboseli for my PhD because much is known about the behavior, the ecology, the demography, and the genetic and social structure of the baboons at this site. And I was therefore able to study topics in the baboons at Amboseli that I would not have ordinarily been able to study in other populations of wild primates. So for my PhD, I studied the endocrine correlates and fitness consequences of variation in mothering behavior in the wild Amboseli baboons. I'll be focusing much of today's talk on my work, the work that I did in Amboseli on the baboons there, and then during the last 10 minutes, I want to tell you about some exciting new research that I've been doing in collaboration with Peter Fashing, a fellow faculty member at Cal State Fullerton, on geladas at Gwasa in Ethiopia, north central Ethiopia. In 2003, Jean Altman and her husband Stuart published a paper in Animal Behavior that described how field studies of animal behavior had changed over the last 50 years since the founding of the journal. In their paper, Altman discussed how field studies of animal behavior had gone from studies in the very beginning that were devoted entirely to descriptive natural history to those that include development and testing of explanations for the causes and consequences of behavior today. Altman credit this amazing transformation to the field of animal behavioral ecology to, among other things, the development of a number of tools and techniques, including the development of techniques for remote tracking of animals and for non-invasive field measurement of a range of physiological variables and a range of other behavioral, physiological, genetic, and environmental phenomena in those animals. So I'd like to tell you about 2 projects that I conducted at Amboseli that capitalize on these recent developments, research projects that combined a field-based study of animal social behavior with non-invasive field measurement of physiological variables and genetic relatedness to test explanations for the causes and consequences of variation in animal social behavior. So let me begin with a very brief introduction to my particular area of research, which is mammalian reproductive biology. For animals, mammalian infants are unusually dependent on their mothers for growth and survival, and this is due to two key hallmarks of mammalian evolution: internal fertilization, or gestation, and lactation. Both of these processes obligate female mammals to invest a great deal of time and energy into each offspring. Motherhood and infancy are two of the most important life stages for any mammal, including our own species. And indeed, maternal care is probably the single most significant measure of successful adaptation among female mammals. And this is because variation in mothering behavior can dramatically affect offspring fitness, growth, and survival. And since maternal fitness is closely tied to offspring fitness, the success of the mother-infant relationship can also dramatically affect the mother's own lifetime reproductive success. A major goal of mammalian reproductive biology is to therefore understand the causes and consequences of variation in the mother-infant relationship. I first became interested in this topic in graduate school after reading Jean Altman's book, Baboon Mothers and Infants, which was based on Jean's own thesis research more than 30 years ago on this topic. At the time, very little was known about the role of chemical messengers in initiating or maintaining maternal behavior in any species, and Jean Altman limited her study solely to the behavior of mothers and infants at Amboseli. One class of chemical messengers, hormones, are particularly interesting because they are produced in only certain parts of the body, but they have wide-reaching effects anywhere where they can get to through the bloodstream. And since the 1970s, there has been an extensive body of research on the mechanisms by which hormones ensure successful pregnancy and by which they mediate the onset, maintenance, and termination of maternal behavior in several laboratory species, including rats, sheep, and rabbits. As Jean and Stuart Altman point out in their review in Animal Behavior, the recent development of behavior-friendly, non-invasive methods to measure excreted steroid hormones in animal waste products, which do not require capture and immobilization of animals, has made these approaches more readily available and widely applicable to more species and more populations. And in particular, these methods can now be used on animals which cannot be trapped or darted for blood sampling, or in cases where researchers want to repeatedly sample from an individual, but collecting blood repeatedly can cause distress to the animal involved, like during pregnancy. My research at Amboseli focused on the nature and extent of both internal and external influences on the mother-infant relationship. And I was particularly interested in identifying and measuring the predictors and determinants of variability in the mother-infant relationship and in evaluating the likely consequences of these differences. This project had several specific aims and goals, but today I'm going to tell you about two of the projects that I conducted at Amboseli. First, I evaluated potential hormonal and non-hormonal predictors of variability in the mother-infant relationship. Naturally occurring variation in the behavior of mothers towards offspring can reflect differences in caregiving motivation. And we know from extensive studies, particularly of the laboratory rat, that these individual differences, even among genetically identical individuals, can have huge influences on the offspring. Now, most research in this area has focused on laboratory animals like the laboratory rat, or where human subjects have been used, have relied on self-reports rather than direct observations of mother-infant interactions. For my project, my collaborators and I combined direct observations of the baboons at Amboseli with recently developed non-invasive techniques to measure hormones in feces. And this study design allowed us to evaluate naturally occurring variation in behavior and also to evaluate potential hormonal and non-hormonal sources of variability and also some of the likely physiological consequences of variability in animals in maternal behavior. Now, before I tell you about my specific research questions and results, let me first just briefly go over my study population, field site, and general methods. So the subjects for the study are part of a larger population of wild baboons inhabiting the Amboseli Basin, which is a large semi-arid savanna grassland ecosystem situated at the base of Mount Kilimanjaro in East Africa. The range of the monkeys now extends between southern Kenya and northern Tanzania. The baboon population at Amboseli has been under continuous observation for over 40 years, and data on life history variables are available on for several hundred animals. At the time of my study, there were a little over 200 individuals living in 5 different social groups in the population. Baboons are among the largest, most sexually dimorphic, and terrestrial of the monkeys. They live in multi-male, multi-female social groups, which can number anywhere from 10 to 200 members. At the time of my study, the largest of the 5 groups, had over 80 members in it. Males typically disperse from their group of birth as they near adult size, anywhere between ages 5 and 8, and they may continue to migrate between social groups throughout their lives. Females, in contrast, remain in their group of birth their entire life, and as juveniles, females will actually inherit their mother's dominance rank in the group's dominance hierarchy. This maternal dominance rank inheritance system is also found in a number of other primate species like macaques, and is also found in hyenas, which I guess you guys heard about last week. Most baboons give birth to their first offspring around age 6, and females continue to produce a single infant approximately every 2 years thereafter throughout their adult life. And like humans, baboons show virtually no seasonality in reproduction. Following birth, a neonate is carried almost continuously by its mother, and it is completely dependent on her for nourishment, protection, transport, at least for the first 6 months of life. I studied 34 mother-infant pairs in 5 groups during a 16-month period, and from July 2002 to November 2003. Because pregnancies can be detected visually in baboons, I began observations on all pregnant females 2 months before they were due to give birth, and I continued observing females for 2 months after they gave birth. Using 20-minute focal animal samples, I collected continuous records of all occurrences of social behaviors between focal females and infants, if any were present, and other group members. I recorded the actor and the identity of the initiator and the identity of the recipient of each social behavior, and for some social behaviors that I was interested in duration, length of time that the animals were involved in those behaviors, I also recorded the time of onset and time of termination of those behaviors so I could afterwards determine how long animals were engaged in those behaviors. Observations were balanced for time of day and across the different weeks of pregnancy and infancy. This list provides some information about the mothers and infants included in this study. And as you can see, mothers and infants included individuals of all backgrounds. About half of the infants were males and about half of the infants were female. Mothers also included females of a variety of dominance ranks, including rank 1 females who were the top ranking female in their own group. And my study also included individuals who were pretty low ranking, including this female here who was the lowest ranking member of her group. Females also varied widely in maternal experience. So first-time mothers like Lollipop here, the current infant is her first infant. And then for females like Vinyl 9, the current infant is her 9th infant. So you can see that females varied widely in maternal care experience, prior maternal care experience. I also collected fecal samples from all pregnant females that I was focal sampling before and after those females gave birth. And the purpose of this fecal sample collection was to be able to assay for steroid hormones later on in the laboratory at Princeton. I should say here that my study focused only on steroid hormones because they're the only hormones that are excreted in feces. Both the major classes of hormones, protein and steroid hormones, are excreted in urine, but for a terrestrial ground-living animal like baboons, urine collection isn't really possible, so terrestrial primate researchers like myself have to content ourselves with being able to study steroid hormones in feces. I collected an average of about 1.3 samples per individual, per female, per week for each of the 16 weeks, 8 weeks before and 8 weeks after birth. At Princeton, I measured the concentrations of the metabolites of 4 steroid hormones in each fecal extract using I-125 radioimmunoassay kits that had previously been validated for this population. Those of you who are not behavior endocrinologists will be relieved to know I won't go into any detail about those assays here, but if you have any questions, I'd be happy to go over them at the end of the talk. So in the first of the baboon studies that I'll talk about today, I evaluated the potential sources of variation in the mother-infant relationship. And the literature on motherhood and infancy in mammals is filled with descriptions of important developmental milestones, like when infants first begin to spend time away from the mother. But rather than describe when certain events occurred, I wanted to describe the ontogeny, that is, how the mother-infant relationship actually develops. And 2 sets of behaviors seem particularly important for defining this relationship. One was the mother's response to infants' distress cries, and 2 was how mothers and offspring negotiate how and when offspring would attain independence from mom. Now, these interactions not only meet offspring demand for support, nutrition, transportation, or protection, but may also be instrumental in preparing offspring for life beyond their mother's body. Now, offspring distress cries elicit parental care in many animal species, and these cries are widely believed to represent honest indicators of offspring need or condition. And variation in maternal or caretaker response to these offspring cries are thought to influence, or believed to influence, the outcomes for infants, including infant growth patterns, development, or survival. At Amboseli, I found that baboon mothers varied pretty widely in their response to infant distress cries. Mothers responded to, that is, they positively responded to infant cries about 1/3 to 92% of the time. Furthermore, mothers appeared to be consistent in their response. So more responsive moms, that is those moms who respond to a greater proportion of all the infant cries that her infant generates during those first 2 months of life. Mothers who were more responsive, responding to a greater proportion, also tended to respond faster. The latency to response was much shorter for these individuals, which suggested to me that these 2 behaviors, these 2 different dimensions of response, were actually coordinated in supportive mothers. Differences in maternal response to infant stimuli, like distress cries, so variation in individual variation in response to distress cries can result from differences in maternal age or maternal experience, like parity, physical conditions, social status, like dominance rank. These differences can also emerge due to differences in infant sex or the rate at which infants cry. However, we found that in baboons, variation in maternal response to infant cries was not predictable from any of these usual suspects. And in fact, in other words, individual variation in maternal response was not predictable from maternal dominance, rank parity, or infant sex, or even the rate at which infants cry. And indeed, the only variable that explained a significant proportion of variation in maternal response to infant cries was the level of fecal glucocorticoid hormone in the mother's feces the month before birth. What we found was that mothers with higher glucocorticoid levels the month before birth responded to a greater proportion. So this is proportion of infant distress cries responded to. Mothers with higher glucocorticoid levels the month before birth tended to respond to a greater proportion of her own infant's distress cries. And when we compare to mothers who had lower levels of fecal glucocorticoid hormones the month before birth. And when we constructed a multivariate model incorporating prenatal fecal glucocorticoids and other potential predictors of maternal response, the prenatal GCs predicts maternal response pattern persisted even after we controlled for all these other variables. Now, why would glucocorticoids, hormones secreted as part of the vertebrate stress response, be associated with individual variation in maternal response to infant distress cries? Glucocorticoids are hormones secreted as part of the long-term wing of the vertebrate stress response. The vertebrate stress response is a suite of short-term and long-term behavioral and physiological events that help vertebrates cope with and deal with challenging events or stressful stimuli by mobilizing energetic resources that are needed to overcome these challenging events. In 2000, Robert Sapolsky, the neurobiologist at Stanford, who also happens to freelance as a hugely influential baboon behavioral endocrinologist, speculated that there may be two types of glucocorticoid action. And the first of these two types is one that everyone here is probably very familiar with. These modulating actions, these actions of glucocorticoids that help an animal respond to an immediate current stressor. An animal experiences a stressful event, activates its vertebrate stress response, and as a response, high levels of glucocorticoids are detected in the blood, and these elevations in glucocorticoids will help the animals by providing energetic resources needed to cope with a challenging event. Now, Robert Sapolsky also suggested a second class of glucocorticoid action that had not previously been documented, but he suggested that maybe there are instances where glucocorticoids may help to prepare an animal for a response to a future challenge. Now, Sapolsky had no proof that preparative actions existed anywhere in the literature, but he suggested that wherever there might be predictable, challenging events that occur, like, for example, seasonal fluctuations in food availability — you can see this is a pretty dry habitat right now — or temporal variation in male-male competition for mates. Wherever you have these predictable, energetically challenging future events, elevations in glucocorticoids might help to prepare animals for these anticipated future challenges. For female mammals, the parturition and the postpartum care of offspring represent predictable, energetically demanding challenges. However, no one had as yet tested the possibility that glucocorticoids might help females prepare for motherhood. And our results for Ambacelli suggest that are consistent with the idea that glucocorticoids may help to facilitate increasing awareness or arousal to infant stimuli, possibly by organizing or activating existing neural circuitry that are involved in responding to infant stimuli. The general increase in glucocorticoids in the fetal-maternal circulation towards the end of pregnancy in many mammals is typically considered to be critical for promoting normal growth and development of the fetus. But this same increase can also function to prepare pregnant females for responding to the challenges associated with caring for their offspring. Because mothers and fetuses share a blood supply, and because elevations in glucocorticoids during late gestation are primarily of fetal rather than maternal origin, our results for the baboons at Ambacelli raise the intriguing possibility that parent-offspring conflict may underlie the preparative actions of glucocorticoids on maternal responsiveness to infant distress. In the 1990s, David Haig, the Harvard biologist, proposed that pregnancy may give rise to an evolutionary tug-of-war between a mom and her unborn child over the amount of resources that the child can extract from the mother at the expense of the mother's future reproduction. This primary conflict of interest between moms and infants is expected to lead to disputes over how much the infant can grow and how much it can draw from the mother's reserves. Indeed, evidence from recent experiments with rats and humans suggests that Haig's hypothesis, evidence from recent studies suggests or support Haig's hypothesis. These studies have found that evolutionary conflicts appear to underlie a range of complications and disorders in pregnancy, like preeclampsia and child development. These conflicts between a mother and her as yet unborn offspring can also give rise to high levels of circulating glucocorticoids as mothers approach the end of pregnancy in mammals. Studies of rodents and human pregnancy suggest that most of the circulating levels of GCs in the fetal-maternal circulation in late gestation are primarily of fetal origin. And if baboon fetuses are like rat and human fetuses, then they may be largely responsible for the spike or increase in glucocorticoids in the fetal-maternal circulation during late gestation, which means that they may be responsible for the apparent GC-mediated changes in maternal arousal and responsiveness to infant stimuli after birth. Now, it's possible that the fetus may be manipulating the mother into providing it more resources and more care than she would otherwise give by manipulating the release of glucocorticoids to influence maternal arousal and responsiveness into the fetal maternal circulation. Obviously, additional, more experimental research on other systems is needed to test this hypothesis, but Haig's predictions about the conflict that arises between a mother and her as yet unborn offspring provide, I think, a fascinating area for future investigation. Given that we're on the topic of mothers and infants, I want to briefly touch on a new study I have coming out in Animal Behavior on sex differences in the mother-infant relationship. Now, everyone here knows that many animals exhibit major sex differences in behavior and ecology as adults, but surprisingly few studies have examined sex differences in behavior among immature animals, especially newborn animals. And indeed, we know very little about whether and how much offspring sex influences the manner in which maternal care or caregiving is delivered to the offspring. And in this study, we used data on mother-infant neonatal physical contact to assess the quality of the mother-neonate relationship, especially the timing of infant independence. We also evaluate the extent to which variation in this timing of infant independence is predictable from a number of variables, including prior experience with offspring, maternal dominance rank, infant sex, or levels of fecal estrogens or progesterone. These are the hormones that are typically implicated in coordinating maternal behavior in laboratory rodents. We found that newborn infants of more experienced mothers possessing higher parity numbers, we found that newborn infants of more experienced mothers initiated higher rates of transitions or changes in mother-infant physical contact than newborns of less experienced mothers. In other words, newborns of mothers with lower parity numbers tended to exhibit lower rates of transitions that were initiated by the infant. Now, what was really exciting was that at each level of maternal experience, the line for males is higher than the line for females. That is, at each level of maternal experience, newborn males initiated higher rates of changes in mother-infant contact than newborn females. Yeah?
I don't understand the transition. What is this variable?
This is the rate of changes in mother-infant contact that's initiated by the infant. So the infant is the one responsible for breaking and making mother-infant contact rather than the mother. So the idea here is that these infants that exhibit higher rates of that initiate higher rates of changes in mother-infant contact are more active infants or allowed to.
Going away from their mother or going toward their mother?
No, simply they're infants. They're really young, so they're actually just the breaking of physical contact. They could be sitting right next to her, but they're not physically on her body somehow. So without the sort of physical comfort of being close to her or physically in contact with her.
But it could include transitions back into contact?
Yes, so you can initiate out and into contact with the mother. So I recorded who initiated each type of interaction, whether it was the mother, the infant, which the vast majority of cases it was mother or infant, and then there was a small percentage where it was initiated by someone else being disruptive to the pair. Okay. None of the other variables that I looked at made statistically significant contributions to explaining variation in this behavior, in this rate of transitions initiated by a focal infant. And the results of a hierarchical multiple regression that we conducted indicated that maternal experience or parity explained 34% of the variance in the rate of infant-initiated transitions or changes in mother-infant contact, while infant sex explained an additional 24% of the variance. These results suggest that more experienced mothers and mothers of male infants may either be more encouraging of emerging infant independence, or they may simply be more tolerant of emerging infant independence than less experienced mothers and mothers of female infants. And to our knowledge, this is the first study to provide evidence of the influence of cumulative maternal experience on the mother-neonate relationship. And also, our study is the first to document the emergence of sex differences in the mother-offspring relationship during the neonatal period for a wild primate. our results suggest that the well-documented differences, sex differences in life history, behavior, and ecology in primates and other social mammals may have their origins in the neonatal period. Okay, so I'd like to now tell you about a third study, the last one that I'll be talking about today, on the baboons at Amboseli. Yeah?
So is it the same pattern if you break it down by breaking contact versus making contact?
Breaking contact versus, you mean if I split the data up into breaking, well, what would be the biological reason why you would want to know that?
Like you were making claims about the infant wanting independence or the mother wanting independence, and that's different from just changing direction of contact.
Okay. It's been a few years, but I believe I did look at those variables separately to make sure that neither one was contributing disproportionately to this rate. And so mothers and infants tend to spend a lot of time in physical contact. So if you look at the amount of time they're actually in physical contact, it doesn't really change that much over the first 8 weeks of life. I mean, it does decline, but variability across, say, all 2-week-olds is not that great. But if you look at the changes that they make, my reason for lumping them together was neither of them made a significant contribution to the variable alone, and B, I was interested in just understanding who is the one initiating, taking the initiative to make and break, because to me that says that the infant either exhibits greater initiative or is allowed to exhibit that greater initiative by the mother. So for me, biologically, it just made more sense to keep them together. So, sorry. But that's a good question. All right, so in the third study that I want to talk about today, the third and last baboon data study that I want to mention, for this project, my collaborators and I combined observations of the baboons with recently developed non-invasive techniques to extract DNA from feces. And this was a study that I did to better understand the adaptive value of enduring male-female friendships in baboons. This study design relying on both field observations of male-female friendships and non-invasive sampling and measurement of their genetic relatedness and their relatedness to any potential offspring allowed me to evaluate the possibility that males may be engaging in biparental care of offspring in this multi-male society. Now, close associations between adult males and lactating or non-fertile females and their dependent offspring are rarely observed in mammals, but they're regularly observed in several primate species that live in multi-male, multi-female societies. And over the past few decades, these bonds have been well documented in a number of primate societies and have become to be known as friendships. And I wanted to understand the adaptive value of friendships to both the male and female participants. Year-round male-female associations are generally rare in mammals because males and females differ markedly in reproductive strategy. While gestation and lactation predispose female mammals to invest a great deal of time into each and every offspring, mammalian males can sire virtually an unlimited number of offspring their fitness success or their fitness is solely limited by their insemination success, or so we think. Males are therefore expected to abandon females after fertilization in favor of more fertile females. And indeed, in the vast majority of mammals, this is what you see. Associations between males and females are confined largely to the period of female fertility. However, year-round associations between adult males and females have now been reported in over 2/3 of primate genera compared to just 1/6 of genera in most other orders of mammals. And these associations can serve multiple functions, including joint parental care of offspring, maybe joint resource defense, or mating effort by males, in which a male can advertise his ability or his willingness to care for offspring in exchange for future mating opportunities with the infant's mother or with other females who observe his behavior. And in birds, where male-female bonds are common, and in some rodent species and insects, male-female associations are typically found in socially monogamous animals, exhibiting biparental care of offspring. But in primates, year-round male-female associations are regularly observed even in non-monogamous species. And in fact, highly specific male-female bonds or friendships between adult males and non-fertile lactating females and the dependent offspring have been widely and regularly observed in a number of primate species that live in multi-male, multi-female societies in which the female mates with multiple males during her fertile period. And these friendships typically consist of a lot of spatial proximity and a lot of affiliative and other types and other grooming behavior between the male and these non-fertile females. And the absence of mating exclusivity among friends suggests that males should invest little in infant care. However, the fact that friendships have been reported in many multi-male primate species raises the question of what it is that males and females are getting out of these relationships. And in this study, we evaluate the adaptive value and determinants of friendships for both the male and female participants. And we use data from genetic paternity analyses, patterns of behavior among parents, that is mother, father, and offspring and other group members. And we also relied very heavily on the long-term demographic and reproductive data of the Amboseli Baboon Project. And we did this to address 3 objectives. One was we wanted to describe or examine the patterning of male-female bonds during the early postnatal period in the Amboseli baboon. So just to describe the pattern of friendship that exists in this population. And secondly, we wanted to evaluate whether females gain social benefits from forming these associations with adult males. And then finally, we wanted to look at the male side of the story and evaluate the nature and extent to which friendships may represent, say, joint parental care of offspring or male mating effort in the study population. All right, before I tell you about my results, I wanted to go over just a few things briefly. One is baboons have externally visible signs of reproductive condition, and the bare areas of skin around the perineum swells and reaches its maximum size around the time of ovulation. Most matings occur within the context of mate guarding episodes called consortships, during which time the male and female are in very close proximity, oftentimes scurried away from the rest of the group. And these consortships typically occur during the follicular phase of the sexual cycle. And it's during this phase the females have these very prominent swollen sex skins that reaches their maximum size around the time of ovulation. And then after ovulation, they begin to deflate. Mothers are reproductively senescent or exhibit postpartum amenorrhea for about 10 months after birth, and the interval between subsequent births is a little less than 2 years. And for this study, I focused on the 29 mother-infant pairs that lived in the 4 multi-male groups in the population. And data on grooming bouts, including actor and recipient identities, and the time of onset and termination for each grooming bout were collected during those 20-minute focal animal samples. Also, I should mention that within the 20-minute focal animal samples, I collected scans at 5-minute intervals on the nearest neighbors of the mothers within 5 meters. Every 5 minutes. And I used measures of grooming and proximity between males and females to differentiate pairs with particularly strong bonds from all other pairs. Grooming plays a key role in primate social relationships. It plays a key role in the formation and maintenance of social bonds in primates. And grooming intensity has often been used as an indicator of the strength of affiliation between grooming partners. In addition, spatial arrangements within groups, that is proximity patterns, provide the broad outlines for social opportunities among group members. And spatial proximity is often used as an indicator of preference in mate choice tests. Females in some species even exhibit enhanced reproductive success. That is, they produce better quality offspring when they're mated with their preferred male. Okay, here's what I found. 1, females exhibited consistency in their choice of male social partners. I found that 25 mothers had both a most frequent male nearest neighbor and a most frequent male grooming partner. These were the guys that they spent the most time grooming and being in proximity to. And for 76% of these mothers, the 2 individuals were the same, or the 2 classes of individuals were the same person, or the same male baboon. And for 4 of the mothers were never seen grooming with any adult males. And so for this reason, I decided to consider each mother's most frequent male nearest neighbor as her male friend, due to the congruence or the agreement between the most frequent male nearest neighbor and most frequent male grooming partner, and the fact that 4 of these females did not groom with any males during this period. All right. We found that females spent significantly more time in proximity to their most frequent male nearest neighbor, that is their male friend. They spent more than 2 times as much time in proximity to their male friend than they did to the the male with whom they spent the second most amount of time in proximity to. And for this reason, we chose to assign only 1 male friend to each mother-infant dyad. And we used the amount of time spent in proximity to her friend as a measure of the strength of their friendship. All right, so to reiterate a pattern I just mentioned a few moments ago, I want to restate that in the vast majority of dyads, the mother's most frequent male proximity partner was also her most frequent male grooming partner. And this congruence allowed us to differentiate male-female pairs with particularly strong bonds from all other dyads that was not characterized by these particularly strong bonds. All right. So now that we've identified friendship dyads, let me now move on to examine the adaptive benefits of friendships, first for females, then for males. Friendships with a male may provide females protection for her offspring or herself against various sources of injury or mortality. Mothers and infants are particularly vulnerable to social aggression, which is a pervasive feature of social life in primate societies. And by associating with an adult male, females may gain the male's protection for herself or her offspring from social aggression committed either by members of her own group or members of nearby groups. Social aggressors can include both immature and adult animals, males or females. And in many group-living primates, females, both adults and immatures, are highly motivated to interact with infants. But some of these interactions may potentially be harmful to infants. And in fact, persistent aggressive infant handling and harassment and kidnapping has been found to result in death or injury for infants and may in fact represent a form of reproductive competition among females. Mothers with young infants can reduce their risk of harassment by associating with male friends who may protect or buffer the dyad from intrusive infant handling or other aggressive acts by immature and adult animals. And to examine this possibility following birth, I kept continuous records of, during focal animal samples, of all bouts of infant distress and all occurrences of infant handling by other group members. And I found that adult and immature females were responsible for the vast majority of acts of infant handling. And in this graph, I plotted the rate at which infants were handled by other group members, adult and immature females, adult and immature males, in increasing order of roughness, starting with touching, and then pulling, which is where the mother pulls, the third party pulls the infant away from the mother, and then in cases where the third party is successful in breaking mother-infant contact as a result of pull, we call those breaking. And I also combine pulling and breaking into a single measure called rough handling. And if you compare the number of handling acts performed per hour per potential partner of each age-sex class, you'll see that females of all ages, adult and immature, were much more motivated and active in handling infants, from touching, pulling, breaking to, and including rough handling. So females of all ages were more active in handling infants, and that disparities in handling frequencies across the different handling behaviors were relatively consistent. So on average, infants were handled most frequently by adult females, secondly by immature females, then adult males, and then least often by immature males. I also found that infants whose mothers spent more time in proximity to male friends received less rough handling and cried less often. In fact, mother-infant pairs that spent the most time in proximity to male friends, that is, who ranked in the top quartile of time in proximity to male friends, experienced the lowest, significantly lower rates of rough handling than mother-infant pairs that spent less time in proximity to male friends. I also found that infants whose mothers spent more time in proximity to male friends cried less often. They exhibited fewer bouts of infant distress cries. And in fact, these mothers, both mothers who spent the most time in proximity to male friends, these dyads received less rough handling and cried less often, experienced the lowest rates of rough handling, and exhibited the lowest rates of distress cries. So I found evidence that mothers and infants benefited directly from friendships. Friendships provided mothers and infants protection from harassment from other adult and immature females. This study was the first to find evidence consistent with the hypothesis that by forming friendships with males, mothers may gain direct benefits for their offspring in the form of protection against harassment by other females. And in addition to protection from female harassment, male friends can also offer mothers and infants protection from potentially infanticidal males. And infanticide is regularly observed in a range of primates and non-primate animals, from lions to rodents, and is widely considered to be an alternative reproductive, a sexually selected alternative male reproductive strategy. And by killing unrelated infants, an infanticidal male can accelerate the return to fertility of a lactating female by removing the suppressive effects of suckling on her physiological system. And by removing this source, the suppressive effect of suckling on postpartum fertility, infanticidal males can accelerate the return to fertility of lactating females, thereby increasing the total number of potential females with whom they can mate with, as well as remove potential competitors for future offspring, for their own offspring. So, and in fact, infanticide avoidance has been suggested to be the major driving force behind the evolution of year-round male-female bonds in social animals, including primates. And if male bonds evolved as a counter strategy to infanticide, we would expect that during periods of heightened infanticide risk, for example, during periods like recent turnovers in the number one position in the male dominance hierarchy in the group, or during periods where the group has just experienced an immigration of a new unfamiliar male into the social group. During these periods of potentially heightened infanticide risk, we would expect that mothers with young infants would increase their rate of affiliation with male associates. However, we did not find any evidence consistent with this hypothesis. At Ambicelli, mother-infant pairs did not spend more time in proximity to male friends during periods of heightened infanticide risk. That is, mother-infant pairs did not spend more time with male friends when there had been a recent turnover. One of these lines is months since new adult male entered the group, and then months since alpha male held top-ranking position. So during months when there had been a recent turnover in the number one position in the male hierarchy, or when there had been a recent immigrant to the group, mothers and infants did not spend more time in proximity to their male friend. And it's possible that infanticide may have been the initial selective force driving the evolution of year-round male-female bonds, but in those populations where infanticide risk is presently low, like in Ambicelli, friendships may be maintained by the secondary benefits gained through protection from other dangers, such as harassment from other females. All right, so let me now quickly turn to the males and look at the adaptive benefits of friendships for them. So 34 adult males were resident in the four multi-male study groups during the study period, and of these, 16 males were friends with at least one female, giving a total of 29 friend dyad and 172 non-friend dyad. And the benefits that males can potentially gain from affiliating or associating with particular mother-infant pairs are a little less unclear, or a little less clear. This is because little less unclear. Are more unclear, thank you. This is because opportunities for paternal care, as you well know, are limited in mammals where infants are almost entirely dependent on mothers for support and nutrition. For males, friendships can represent a form of paternal care or male mating effort, or both. The paternal investment hypothesis proposes that by associating preferentially with his own offspring, a male friend will help to improve the fitness of the offspring he has sired with the female. Given that female baboons mate with multiple males during the fertile period, we would expect male baboons to play only a minor role in infant care. However, emerging evidence from a number of other studies from a number of other species show that males, in many instances, can and do recognize their own genetic sire, even when there are offspring in the brood that they are caring for that are not their own genetic offspring, that they actually make these distinctions. And to evaluate the paternal investment hypothesis, we use data from genetic paternity analyses to assign paternity for 78% of all individuals present in the four groups during the study period, including 80% of the immature animals that were present in the study groups at the time. And lucky for me, 23 of these 80% of immature individuals, 23 of these immature individuals were infants during my study and had fathers who were present in their group during the first 2 months of life and were parts of the study. We also use data recorded during mating events and mate guarding episodes to identify all adult males who might have sired the offspring. These are males who consorted with the mother during the 5 days of most likely conception of that offspring during the cycle in which the mother conceived that offspring. And we call these guys behavioral fathers as opposed to genetic fathers. All right, here's what we found. Nearly half of all male friends were the genetic fathers of offspring and had been observed mating with mothers during the last days of most likely conception of that offspring. Now, in contrast, nearly all friends who were not fathers were also not observed consorting with the mother during the days of most likely conception of the offspring. And fathers who were friends were significantly more likely than friends who were not fathers to be present in the group during the mother's future reproductive cycles. In other words, friends who were fathers were more likely to stick around than friends who were not fathers. So, could friendships at Ambicelli represent a form of paternal investment in offspring? And the results of our study suggest that many, in fact, about a little more than half of all friendships represent a form of paternal investment in offspring. Our results also provide intriguing evidence that paternal kin recognition and investment in offspring may begin immediately after birth, because nearly half of all friendship dyads consisted of a mother-infant pair, of mother-father pair. And our results are consistent with some exciting new data. Our results are consistent with some exciting new data from studies of birds, fish, and several primates, including Ambicelli baboons, that suggest that males in many instances, can distinguish their own juvenile offspring from unrelated ones. This study happened to focus on much younger individuals. Intriguingly, nearly all friends who were not genetic fathers were also not behavioral fathers, that is, males who had consorted with the mother when she was most likely to have conceived that infant. This finding indicates that friendships between non-fathers and mothers in this population were not simply the result of paternity confusion. That non-father friends were not misled by past mating behavior into forming friendships raises the question of what actually, these are some examples of males that can and do recognize and distinguish their own juvenile offspring from unrelated offspring. All right, so did I go back in time? Did I just go back in time? Yes, I did. Okay, sorry. Oh, I didn't, okay. Sorry. So nearly all friends who were not genetic fathers were also not behavioral fathers. So the fact that these guys were not misled by past mating activity raises the question of what drives them to become friends, what actually motivated them and rewards them for becoming friends. Now, in addition to representing paternal investment in offspring, friendships could instead or also represent a form of mating effort by males. That is, male care of infants may function to increase their attractiveness as a potential mate to the mother's infant or to other females who may see them behaving as a good father figure. This idea, called the mating effort hypothesis, proposes that by demonstrating their willingness to invest in an infant, male friends may enhance their chances of being chosen as a partner, a mating partner by the female or other females nearby in future reproductive cycles. So did male friends at Ambicelli, regardless of whether or not they were fathers, achieve more mating success with females once those females resumed cycling than what was expected given their average consort activity with all other adult females in the group, which is of course a function of their rank? And the short answer, I can go into a long answer, but the short answer is no. We found no evidence that prior friendship increased a male's chances of mating with a female in future reproductive cycles. We found that friends who continued to reside in the female's social group after she resumed cycling did not achieve greater consortship success with her than they would have achieved otherwise. So contrary to reports from some other populations, friendships in the past did not predict a male's consorting success with a female in the future. And if prior friendship with a female does not increase a male's chances of mating with her in future reproductive cycles, what other benefits can be obtained by the male who is not the infant's father? And this is a big mystery as far as I'm concerned. At the moment, we don't know the answer, but it's possible that males may trade protection for the infant with other services or benefits not yet identified. So I just told you about two studies that took three studies that took advantage of recent technological developments to gain deeper insights into the behavior of baboons and ambacelli. Now I want to just turn very briefly to my new research on gelada monkeys in the next few minutes. So currently my research on geladas focuses on the behavior and ecology of a wild population of this rare and enigmatic primate, which is a close relative of baboons, at a remote field site in the Ethiopian highlands called Gwasa in north-central Ethiopia. The site is located about 3,500 meters above sea level on the western edge of the Great Rift Valley, which you could see in the background. Geladas occupy a higher altitude and colder climate than any other African primate, while the ambacelli baboons of my thesis research occupy a sweltering semi-arid ecosystem at the base of Mount Kilimanjaro. In December 2005, while I was still writing up the results of my PhD research, along with Peter Fashing, I established a research site at Gwasa, and we began the arduous process of habituating the geladas at the site to our presence. And since January 2007, we have monitored the demographic, social, and reproductive status of roughly 175 animals in 15 one-male units on a near-daily basis at Gwasa through our own efforts and with the aid of volunteer field managers and our local field staff. Our research at Gwasa would not be possible without the help of the 10 volunteer field managers who have helped us to date, recent college graduates who each volunteered to spend a year at Gwasa collecting data for the project, keeping things running while we are both away at camp. Nearly all former field managers are now happily enrolled in master's or PhD programs, and this has been a very satisfying aspect of the project. Now, I could tell you a bunch of interesting things about geladas, but in the interest of time, I'll just briefly mention that geladas are unusual among primates and that they are the only grass-eating primate, and they live in complex multi-tiered societies. So geladas exist, this complex multi-tiered society consists of the basic unit, which consists of a one-male unit, usually consisting of a single adult male, and several, anywhere between 1 and 10 adult females. There are 4 adult females here, and their dependent offspring, though sometimes we have follower males as well. One-male units that share a common home range are called a band. The one-male units belonging to the same band don't range together every day. And oftentimes what we are observing a herd of animals, and members potentially from 2 different bands that are just aggregating together. And these herds can number anywhere up to 600 animals at Gwasa. In the Simien Mountains, they can number up to 1,000 animals. All right, in the interest of time, let me tell you about a project that we've been working on recently. I mentioned earlier that geladas occupy a higher altitude and colder climate than any other African primate. High altitude climates tend to be incredibly seasonal, that is characterized by recurrent fluctuations in rainfall and other climatic variables, and Gwasa is no exception. This is a day when daylight, when heavy fog has descended on us. So we would expect that animals that occupy seasonal habitats like geladas would have to cope with these predictable fluctuations in food supply by varying what they eat and how they eat it. And these responses are in turn are expected to influence or impact female reproductive decisions and life histories. So we looked at birth seasonality. We examined patterns of within and between units, that is population-wide birth seasonality in relation to rainfall and primary food productivity in our 15 gelada units at Gwasa from January 2007 to July 2001. And given the extreme climatic conditions at Gwasa, we did expect the geladas to be incredibly seasonal with respect to reproduction. And here it is. All right. This circular histogram depicts the distribution of births across all of our study units across an annual cycle. So from January all the way through to December. This is data from January 2007 through July 2011, and concentric circles represent increments of 3 births, with most births occurring through July and December, based on the sort of length of these lines. Using circular statistics, we found that this population of geladas reproduces seasonally with a protracted birth peak lasting approximately 6 months. And in circular statistics, the parameter R This variable here, the mean vector length, provides an objective measure of the extent to which the frequencies of a particular event are uniformly distributed across a circle. And the parameter r is 0 when all births occur uniformly across the annual cycle, and 1.0 when all births occur at the same time. And for geladas, we found that 69% of the 99 births recorded during this period occurred from July through December. So we have a pretty seasonal primate. The mean month of birth was October here, and we found that the birth peak coincides with the wettest months of the year. That is, July and August are the months receiving the most rainfall. Most of our rainfall occurs during these 2 months. We get about anywhere from 1,500 to 2,500 millimeters of rain a year. That's a lot of rainfall. Most of that rain falls in July and August. The birth peak at Guasa also coincides with the greenest months of the year. And we looked at food availability using 2 measures. One was the total green grass biomass, or as you can see here, this is the green grass to brown grass ratio. And you can see that the birth peak coincides or overlaps with this ratio really nicely. And the birth peak at Guasa appears to be timed to ensure high food availability during late gestation and early to mid lactation. And our results suggest that the geladas at Guasa are pursuing a seasonal breeding strategy that maximizes maternal survival by minimizing energetic stress on a nursing mother. And this is what Charles Jansen and Jennifer Verdelin, in their 2005 meta-analysis of primate birth seasonality in relation to climate, would call an income to or maternal survival breeding strategy. This breeding strategy is found in capuchins, it's found in snub-nosed monkeys, and now geladas at Guasa, and primates that typically occupy highly seasonal habitats. So in short, we see that female geladas at Guasa appear to time birth so that the most energetically demanding phase of reproduction, that is lactation, coincides with yearly peaks in nutrient or energy availability. And we anticipate that the extreme seasonal conditions at Guasa will prove to influence many other aspects of the lives of the geladas at this site. However, as the climate continues to warm, animals like geladas that occupy high altitude mountainous habitats, which are incredibly sensitive to warming, may need to make adjustments to their daily routines to simply stay alive. And we're really just beginning to scratch the surface of the data set that we have been collecting at Guasa since 2007. And we're really excited to continue the research and community conservation activities that we started at Guasa in 2007. Thank you for listening. And in closing, I want to thank the following people and organizations for their help in making my dissertation research and current research on geladas possible. And be happy to take any questions and direct you to our project website if anyone would like more information about our project. Thank you.